So on your screen, you can see a scientist. His name is Hans Adolf Krebs. Born in 1900, a German-American biochemist who is considered as a pioneer in the study of cellular respiration. His remarkable survey of energy transformations in living matter, published in 1957 in collaboration with H.L. Kornberg, which discusses the complex chemical processes which provide living organisms with high energy phosphate. He discovered something for what he got Nobel Prize in Medicine in 1953, shared with Fritz Lipmann. His discovery was the Krebs cycle. In our last video, we talked about glycolysis. Glycolysis एक process था जहाँ पर glucose was converted into two molecules of pyruvate. And this happened inside the cytoplasm of a cell. If you recall that there are three steps in cellular respiration. First was glycolysis, the conversion of glucose into pyruvate. The further conversion of pyruvate comes under the second process, the Krebs cycle. Finally, the last step, the oxidative phosphorylation reactions. The pyruvate formed during glycolysis enters inside the mitochondrial matrix and get converted into acetyl coenzyme A by the addition of coenzyme A in the presence of an enzyme called pyruvate dehydrogenase complex, or PDC, accompanied by the removal of hydrogen as NADH, and CO2 is removed also. Thus, this reaction is termed as, more accurately, oxidative decarboxylation, but vaguely it studied as pyruvate oxidation, a step prior to the Krebs cycle. Krebs cycle, also known as TCA, Tri Carboxylic Acid Cycle, is also known by yet another name, which is Citric Acid Cycle, because citric acid is the first stable compound to be formed in this cycle. Here is the place where Krebs cycle actually begins. So here it begins, the Krebs cycle. This is the detailed description. Acetyl coenzyme A, in the presence of water, is condensed with oxaloacetate to generate the first stable intermediate or substrate, which is an important one, named as citric acid or citrate. Hence the name citric acid cycle. This is the first process in this cycle called condensation, which is catalyzed by an enzyme, citrate synthase. Now, in second steps part A, which is a dehydration reaction, water is removed from citrate to form cis-aconitate. This dehydration is followed by the part B of step 2, which is an antagonistic reaction to A, i.e. a hydration reaction forming isocitrate. So this two-partite reaction of consequent dehydration and rehydration is catalyzed by an enzyme known as aconitase. Now, in the next step, means the third step, named as oxidative decarboxylation, which is marked by the removal of carbon dioxide and hydrogen to form alpha-ketoglutarate. This reaction is catalyzed by isocitrate dehydrogenase enzyme. Next, the step fourth. This is another oxidative decarboxylation, but this time alpha-ketoglutarate is subjected to this process. The only difference here lies in as the addition of coenzyme A, which consequently form succinyl coenzyme A and another substrate. This reaction is catalyzed by alpha-ketoglutarate dehydrogenase. Moving on to the next step, step 5, marked by the removal of coenzyme A, and here ATP or GTP is formed. That is why this step is known as phosphorylation, catalyzed by succinate thiokinase enzyme. And in this reaction, succinate is formed. Let's talk about the next chemical reaction, where FADH2 is formed in this sixth step, which is called dehydrogenation, catalyzed by an enzyme called succinate dehydrogenase, forming fumarate as a result. Next, water is added in seventh step, that is hydration, catalyzed by an enzyme called fumarase, forming malate consequently. Now, finally, malate is converted back to OAA, oxaloacetic acid, with the removal of hydrogen as NADH. This is the final step, step 8, called dehydrogenation, catalyzed by an enzyme called malate dehydrogenase, yet another dehydrogenase enzyme. And after oxaloacetate is formed, condensation with acetyl coenzyme A and all the consecutive steps performed in the cyclic manner again in the Krebs cycle. And the cycle goes on and on. That was the detailed description of Krebs cycle, where various intermediates or substrates formed in a cycle to form some energetically important outputs such as NADH, ATP, GTP, and FADH2 at several steps. Finally, a mnemonic to remember the names of substrates in Krebs cycle. This mnemonic goes like: citrate is Krebs' special substrate for making oxaloacetate. Now, let's understand this. Citrate for citric acid is for isocitrate. Krebs for alpha-ketoglutarate. Special for succinyl coenzyme A. Substrate for succinate. For fumarate. Making for malate. And finally, the oxaloacetic acid or oxaloacetate OAA. This was the mnemonic to understand or to remember the different substrates that comes in the cycle known as Krebs cycle, also known as TCA or Tri Carboxylic Acid Cycle. This is called so because the first product formed in this cycle, which first stable product formed in this cycle, is a citric acid, which is a tricarboxyl acid or tricarboxylic acid because it contains 3 carboxyl groups, COOH, you can see in this structure of citric acid. The overview of Krebs cycle. The Krebs cycle results into the formation of 3 important compounds. The first is NADH, which are formed at every oxidative decarboxylation reaction. And there are 2 oxidative decarboxylation reactions in the Krebs cycle. Besides NADH, there is the formation of ATP or GTP, and finally, there is the formation of FADH2. So, in this whole cycle, the resultant compounds that are important for the further reactions are NADH formed in the step 3 and 4 as a result of oxidative decarboxylation. ATP or GTP formed in the step 5, which was a phosphorylation step. And FADH2 is a product which is formed during the step 6, the dehydrogenation reaction. In Krebs cycle, there are 2 oxidative decarboxylations takes place in step 3 and 4. Besides these 2, one more such reaction occurs yet in the mitochondrial matrix, but this one is not included in the Krebs cycle. This occurs in the presence of PDC, and the step and the reaction is called the oxidative decarboxylation. This is the overview of Krebs cycle. Now, the overview of cellular respiration is a matter of talk. What is now left? But for that, we must walk through the final frame of the picture, named as the oxidative phosphorylation. And that is a topic, a concept for another video. Let's meet in another video. That is all for this one. Thanks for watching. Remember,